So, as you recall, we learned last class about the actual skeletal muscle cell, which is what we're going to be spending most of our time on in this chapter discussing. And so you learned that the skeletal muscle cell has a lot of the components that we learned about in chapter 4 when we were talking about the anatomy of cells. For example, it has a nucleus. Actually, it's multinucleated. It has sarcoplasm, which is the cytoplasm. It has organelles like mitochondria and whatnot. So we're going to kind of delve into a little bit about mitochondria here. And we're going to learn because skeletal muscle is what we would refer to as a highly metabolic tissue, right? Which means it's going to be doing a lot of things. So it needs a lot of fuel, and therefore it's going to create waste and whatnot. And so we're now going to discuss how your skeletal muscle cell makes energy. And so you learned about the mitochondria back in chapter 4. What was the nickname for the mitochondria? Y'all remember? Yeah, the powerhouse. It's the powerhouse of the cell. And its job is to generate, all right, ATP. As you know, ATP is the fuel source that cells use. So mitochondria, right, will make the ATP, and it's not the only structure that makes the ATP. So here's the basic gist of it. If you have oxygen available, you can make a lot more ATP than what you would be able to make if you didn't have any oxygen available. So if you don't have oxygen available, when we're going to create and make ATP, we call that anaerobic respiration, cellular respiration. If you do have oxygen available, then we're going to engage in what we call aerobic cellular respiration. And so if we're going to take one glucose molecule, and if you don't have oxygen, you can only get 2 ATP out of that. Not bad, but it's certainly not very good. If you have oxygen available, then that one glucose molecule can yield you about 32 ATP. See the big difference there? All right, so that's why we can see the value of oxygen. And that's why we want to ensure that your cells are engaging in aerobic cellular respiration. And so what allows our muscle cells, okay, to make all of that energy is the fact that they have tons of mitochondria. And those mitochondria are going to want to make ATP through aerobic means with oxygen. So what brings oxygen to your cells? What system does that? Your blood. Yeah, the circulatory system, right? And we learned last class that skeletal muscle is highly vascularized, which means it has a lot of blood vessels going to and from. So we're already now starting to set our skeletal muscle up for success because we're giving a lot of blood vessels so it can transport a lot of oxygenated blood to these tissues so we can use that oxygen for aerobic cellular respiration so we can make lots and lots of ATP, which we will need when we're using these tissues quite a bit. Well, there's another molecule that we're going to discuss, and that's myoglobin. What's that similar to? What do you think? You've heard of something else that has a name similar to myoglobin. Myo means muscle. Hemoglobin. Yeah, hemoglobin. Right? Hemoglobin is a protein molecule that sits in your red blood cells that allows for the transportation of oxygen. And so in our muscle cells, we have myoglobin, myo being muscle. So it is the muscle's version of hemoglobin. And so this protein attaches oxygen. So you can store oxygen in muscle cells up to a certain amount or limit. And so that's what myoglobin does. It is for stored oxygen. So we can use that oxygen to make ATP aerobically. Okay, so that's good. We've set ourselves up for success there. Well, guess what? We're going to take it a step further. Y'all remember back in chapter 2, we talked about this molecule called glycogen. Glycogen is the stored form of glucose. We're going to take a glucose molecule and attach it to another glucose molecule and attach another one, another one, another one. We call that a polysaccharide. And so that's essentially what glycogen is. It's just a whole bunch of glucose molecules strung together. So it's stored inside the cells. It's part of the cytoplasm. Remember, the cytoplasm has 3 things. Cytosol, which is the fluid component, and then you have the organelles, and then you have inclusions. Inclusions are temporary. That's where glycogen falls into. It's an inclusion because glycogen stores come and go depending on what the cell is going to be doing. So we use glycogen, all right, for the fuel to make ATP aerobically. And guess what? Our myoglobin helps us to do that because it has that stored oxygen there. That's cool. And then we've got another thing that we're going to add into here called creatine phosphate. Creatine phosphate is this molecule, right, that has a phosphate molecule attached to it. And phosphate is one of the ingredients to make, it's on the board here, ATP. Okay, so now we have a molecule that has phosphate attached to it. And so when we use ATP for energy, we're essentially just going to snap off one of the phosphate molecules. And it's usually the third phosphate molecule. So we snap that off. The bond that holds it together on that molecule is energy rich. So when you snap it, it creates energy. So we can do work. So now ATP, when we snap off that one phosphate molecule, is one phosphate molecule short. So now we call it ADP. Well, what we can do is this creatine phosphate molecule will lend its phosphate to ADP to make more ATP. It's kind of cool. I'll get into more details on this later on when we get into, all right, the actual metabolism of a skeletal muscle cell. But I wanted to introduce you to this since we're on the level of that microscopic anatomy there. So again, this slide here is just showing you, right, myoglobin, obviously, is going to be unique to muscle tissue, hence the name myo, right? And it's going to bind oxygen. So that's essentially what's going on. So if you go and work out and then you start to lay down or sit down and rest for a little bit, okay, myoglobin is going around throughout the cell and it's just soaking up and grabbing onto oxygen and replenishing its oxygen stores. So the next time that you start to use that muscle tissue, it's got oxygen to make ATP aerobically there. Right? And so again, when you start to exercise, that oxygen pops right off of the myoglobin, goes into the mitochondria, and allows for our ATP to be made, all right, much more abundantly. Instead of just getting 2 ATP from one glucose molecule, now we can get 32. All right. So what are the primary components of thick and thin filaments? This is from last class. All right, the primary components of thick. So what's the name for thick filament? What do we call that? Myosin. Okay. Thin filament is going to be actin. Okay. So when we talk about the thick, that's the myosin. When we talk about the thin, okay, you have 2 types of actin. All right. I'm not concerned about it, but I am concerned about the other 2 proteins. We call them regulatory proteins that sit on top of the actin molecule. Remember I showed you the video with calcium coming and floating along and settling in on the one molecule, and it causes the other molecule to shift off the myoglobin, or not myoglobin, excuse me, the myosin binding sites there. That's what we're talking about. Thin filament is going to be the actin protein, and we're going to include tropomyosin and troponin. Those are the regulatory proteins there. Because when the muscle's at rest and it's not contracting, right, these binding sites are blocked by tropomyosin. And so when calcium is present and there's ATP, all right, available, then we can get a muscle contraction, but it starts off with the calcium settling in onto the troponin. And then once it does that, it causes the tropomyosin to shift off of the myosin binding sites on actin. All right, and that's in that video, which guess what? We're going to watch again later on. All right. I'm going to have you watch it so many times until your eyes are bleeding. No. Okay. So that's a little extreme. All right. But again, you will see as we move through this chapter, you'll understand that video. And something that may have seemed so difficult, maybe it doesn't seem difficult, but once you go through it, you're like, I understand each and every single one of those points. Okay. So this is now, we're getting into the good stuff, right? This is the part that I really get excited about. All right. We're going to now discuss a skeletal muscle contraction. Okay, might seem sort of mundane to some of you, but you're going to see everything that is involved an action that needs to happen for a contraction to occur. And so we have three things that occur. Okay, we're going to see something occur at the neuromuscular junction. All right, then we're going to see what happens when we get a nerve signal that's going to travel along the sarcolemma which is the plasma membrane of the skeletal muscle cell. And then finally, we're going to talk about what we call the cross-bridge cycling, the interaction between the actin and the myosin and how we shorten those sarcomeres. Right, and so that's the stuff that we're going to be discussing. Not all of it today. It's a lot to unpack, but we're going to get into it. We're going to at least do the first one, what happens at the neuromuscular junction, and then we'll talk about the excitation-contraction coupling there. All right, so let's go over some of the components here of what the neuromuscular junction is. So if you look at that word, obviously it's a compound word that's made up of two terms. Neuro meaning neuron, okay? Muscular meaning the muscle. So we're going to see what is going to happen at that junction, that area where the neuron synapses onto our muscle, our effector. We're going to discuss that. Right, so that means we need to have a couple of things involved. One, we need a motor neuron, not a sensory neuron. Remember, there's two types of neurons. Okay, we need a motor neuron. Then we're going to need a muscle fiber, aka muscle cell. And then we're going to need a space, a synaptic cleft between those two structures there. Okay, so I'm going to assume that y'all watched that video, that last part of chapter 12, about the transmissive segment of a neuron. That's the synaptic knob. And if you didn't, don't you worry. We're going to go over some of the concepts there of what happens at the synaptic knob. Okay, so the synaptic knob is that structure that sits at the end, the distal end of the telodendra. And so you'll see it looks like a big swelling. So if I go back to this picture here, this is it. That's the synaptic knob right here. And so inside the synaptic knob is the neurotransmitter, which is stored in the synaptic vesicles. So it's stored up in there. So for skeletal muscle cells, the neurotransmitter is always, you need to know this, is always going to be acetylcholine. Right? And acetylcholine is always excitatory. It can't inhibit a muscle cell. It's always going to try to stimulate it and excite it. Right? So you need to know those things. Right? So the synaptic vesicles are filled with acetylcholine that are going to be released right from the neuron, diffuse across the synaptic cleft, and then it's going to bind onto chemically gated channels on what we call the motor end plates. That's the area where the synaptic knob kind of hovers over on the muscle cell. And it's going to release its neurotransmitter. It's going to bind onto those receptors there for those channels, and it's going to trigger those channels to open up, and we're going to actually create an action potential, much to what we kind of learned before in chapter 12 here. So what else is going to be on our synaptic knob? Well, you got the calcium ion pumps. Right, the calcium ion pumps have one job, and that's to pump calcium continuously and repeatedly out of the synaptic knob. Because guess what that does? Well, what did the sodium potassium ion pumps in the neuron do? They made sure that there was more sodium outside the cell and more potassium inside the cell. Banana floating in the ocean. It made sure that there was a concentration gradient. So when we opened up channels, it ensured that sodium would enter into the cell and that potassium would exit the cell. Same thing here. Okay, our calcium channel ion pump is going to pump calcium out of the neuron. So it's going to make sure that there's more calcium outside the neuron than inside the neuron. So when we open up these transport proteins, voltage-gated calcium channels, where's calcium going to go? Into the cell. That's right. That's right. That's what we're going to see. It's going to go into the cell. Why? Because it's going to flow down the concentration gradient. I'm pretty confident that y'all kind of understand that to a certain degree. If you made it through chapter 12, right? You understand how that works again? Okay. Calcium ion pumps pump calcium out. So there's a lot of calcium outside the cell, hardly any inside the cell. Then the action potential comes strolling along, rolling down, right? The membrane, the plasma membrane of the neuron, and it activates and stimulates the opening of these voltage-gated calcium channels. So when they open up, calcium then enters into the cell. And then I'm going to tell you what happens then, because now we're going to start messing around with the neurotransmitter. Okay, so this is basically explaining to you what I just said. So the nerve signal or the action potential travels down the axon. It comes in contact with that first voltage-gated calcium channel. That voltage-gated calcium channel opens up. Calcium moves down its concentration gradient, meaning it moves into the synaptic knob. And then the calcium is going to start to bind onto or cover the surface of the synaptic vesicles. I love this part. What type of ion is calcium? It's a cation, right? Positively charged. When you're looking at the membrane potential of a neuron, the charge outside the cell is usually positive, right? And the charge inside the cell is usually negative, right? And remind me again what the charge of calcium is again? Positive. So guess what's going to happen? Opposites attract, right? So when calcium pretty much covers these synaptic vesicles, it gives it a positive charge. Well, guess where those synaptic vesicles are going to move towards? The inside of the plasma membrane. They're going to gravitate towards the inside of the plasma membrane, fuse with the plasma membrane through the process of exocytosis, and then it's going to spill out its neurotransmitter into the synaptic cleft. That's exactly what we're going to see here. So those calcium ions bind onto the surface of the synaptic vesicles. Synaptic vesicles then merge with the cell membrane. Okay? Exocytosis, that's the name of that process. If you don't know it, learn it. Okay? Exocytosis, exo means exit. Right? So if it's going to exit out of the cell, we're going to eject it into the synaptic cleft there. So we're literally going to get thousands of acetylcholine molecules getting ejected out of the synaptic cleft, and they're going to diffuse across the synaptic cleft towards the plasma membrane of the muscle cell. So this is the action. This is what happens, right, at the neuromuscular junction. Y'all kind of see that? And there's pictures here. I'll show you some pictures. Okay. So the neurotransmitter diffuses across the synaptic cleft, and it is going to then bind onto these acetylcholine receptors that are attached to our transport proteins on the muscle cell. And so those transport proteins are cation transport proteins, which means they're going to start opening up chemically gated sodium channels and chemically gated potassium channels. But again, we all remember banana floating in the ocean. We know, I don't even have to go over it with you, but I'll do it. Right? We know which direction that those ions are going to go once those channels open up. Sodium is going to go where? It's going to enter into the cell because there's more sodium outside the muscle cell than inside the muscle cell. Same thing as a neuron. Where's potassium going to go? It's going to leave. Yep, it's going to exit out. Okay, but keep in mind, a lot more sodium is going to go into the cell than potassium leaving. So we're going to make the inside of the muscle cell more positive. What do we call that? When we make the inside of a cell more positive or less negative? Depolarization. So we're going to start to depolarize our skeletal muscle cell, much like we had to do with the neuron. Okay? And just like we had to do with the neuron, we needed a threshold value. What's the resting membrane potential for a neuron? -70. You all got 1 point on your test already, all right? That's going to be a test question. Do y'all remember what the threshold value was? -55. Good, okay. So negative 55 is more positive than negative 70. So that process of depolarization gets us to that threshold value. We're going to see the same thing here. We got some different numbers here. We'll see those here in a second. I don't want to get ahead of myself, but it's the same concept, y'all. Okay. We need to get to that threshold value so we can elicit and create another action potential. And we'll get there. We'll get there. Okay. Any questions about that so far? Not too bad. Right? If I'm moving too fast, slow me down. Best way to do that is ask me questions. All right, so the motor end plate, that is on the cell itself, right? And you'll see it. It's right on the sarcolemma. So you'll have like a relatively smooth part of the plasma membrane, and then you see this little indentation that has these little kind of folds here, and then it straightens out again. And so the synaptic knob hovers above that. And so in those folds there, that's where you're going to see those cation channels, those chemically gated sodium channels and those chemically gated potassium channels, right? They're in those folds. And the reason why we have folds like that is because we're increasing the surface area. So we can jam in more channels in there. That's the reason. So when that neurotransmitter diffuses across the synaptic clefts, keep in mind, it's a fluid-filled space, hence the process of diffusion, right? So it then will bind on to the acetylcholine receptors on those channels. Now, when we stop a contraction, right, we got to clean up the mess. We just threw a party. That's essentially what just happened. We threw a party in the neuromuscular junction. We released all this neurotransmitter. It's like New Year's Eve when they released the confetti in New York City. Well, someone has to clean that up, right? And believe you me, they do it really fast. I've been there. Okay. I've been there at 4 o'clock in the morning and I've walked out of a club at 6 in the morning. It was all cleaned. So they're impressive there. Anyways, so what you're going to see is we got to do the same thing with that acetylcholine. We got to get it out of that synaptic cleft for the next action potential, for the next muscle contraction. So you've got this awesome enzyme here. Anytime you see a word that has these 3 letters at the end of it, ASE, that is how you spell an enzyme. So acetylcholine esterase is an enzyme, okay? And it breaks down acetylcholine. So when it breaks down acetylcholine, it breaks it down into acetyl and then a choline molecule. And then we're going to ship it back into the neuron and we'll fix it and we'll repackage it, combine those 2 molecules together to make acetylcholine. So we're going to break it down in the synaptic cleft to stop everything. And then we're going to use like a vacuum cleaner and suck it all up back into the neuron. We'll stick those 2 molecules back together to make acetylcholine, stick it back into the synaptic vesicles for the next muscle contraction there. All right. So keep in mind that enzyme there, acetylcholine esterase, that's the one that's going to break down acetylcholine there in the synaptic cleft. So here's a great picture showing you everything that I just talked about. So here you can see, let's go to the neuron. Here is the, where are you? Here is the calcium ion pump right there. What's its job? Pump calcium where? Into the cell or out of the cell? Out of the cell. Perfect. That sets up our concentration gradient. Okay, so our nerve signal comes down. It opens up these voltage-gated calcium channels. What direction does calcium want to go? In. So calcium diffuses into the cell. It starts to cover the surfaces here of our synaptic vesicles. That positive charge pulls it towards the negative polarity charge here on the plasma membrane. And they will then fuse here with the plasma membrane and eject its contents out. That process is called what? Exocytosis. Exit the cell. Right? Acetylcholine then diffuses across the synaptic cleft here, and it'll bind on to these cation channels, and it causes them to open up. Sodium can then enter into the cell. Potassium can then exit out of the cell, thus starting to depolarize our cell, approaching the threshold value. We'll get to that part here. Kind of see that a little bit, okay? We just covered 2 chapters right there. We knocked out the transmissive segment in chapter 12, and we just started off, excuse me, what happens at the neuromuscular junction there. All right, so this picture here shows you all those steps. I won't go through them because I just did. Right, but you can see this is figure 1010. So what I would recommend is learn that figure, memorize it, okay? And if you understand it, then you understand the first part of the beginnings of a muscle contraction. All right? All right, anyone ever hear of our first clinical view tonight is Myasthenia gravis? Anyone ever hear of that? All right, it's a bummer of a condition. Again, we're dealing with an autoimmune disease here. So we see more commonly, all right, for those of you that are going into the healthcare field, you will see more commonly women being afflicted with autoimmune disease. My personal opinion on that is one, there's a genetic component, but also women's immune systems in general are better than men's. And that's why. Again, that's my own personal opinion. Right? I'm always sick. My wife and daughter are never sick. And believe it or not, does anyone here know anybody that's never had COVID? Okay, there's a few. You never had? You never got it? Yeah. My daughter never got it. So I know there's folks out there, and I don't know what that is. If there's a study out there, you know, they ought to figure it out, because I'd love to know why some people got it and why some people didn't. You never, yeah, see? And you got, and then again, I wonder if it's a strain thing or whatnot. I mean, there's so many questions I have. And especially, y'all, when we get into chapter, right, what myasthenia gravis is. So essentially what you'll have happen is that your plasma cells are going to generate these antibodies that are going to attack the acetylcholine receptors that we just learned about, that are at the neuromuscular junction. And so they start to attack these receptors, thereby compromising the ability for those receptors to trigger the opening of those chemically gated, all right, channels that we just saw for sodium to enter into the cell and for potassium to leave the cell. So it takes a lot more to get, all right, an action potential to be generated on a muscle cell when you have fewer gates that you can effectively open and close there. So here's what we're going to see. It's harder to stimulate the muscles. So if it's harder to stimulate the muscles to contract, people will complain of primarily muscle weakness, but they'll feel tired faster. And we see it in the smaller muscles. People will complain about having weaker grip. They're more fatigued partially because they can't inhale and exhale as easily. The skeletal muscles of the diaphragm and the external intercostal, I know we talked about this in lab, those are the muscles that are responsible primarily for inhalation during quiet breathing, right? Well, it takes a lot more. It's more difficult to respire, okay? And so they'll have less oxygen, and so they'll fatigue quicker, okay? So there is a drug that we will use on, I can't remember the name of it, right? But there is a drug that we will use on folks to help out with myasthenia gravis. And that drug goes after this enzyme. So if we have a drug that pretty much prevents that enzyme from working, that means we can't break down the acetylcholine that's bound onto those receptors. So that allows those gates to stay open longer. It allows more sodium to enter into the cell, more potassium to leave the cell. So that's one of the solutions for myasthenia gravis. There's probably newer stuff that's out there. Okay, so what triggers the binding of the synaptic vesicles to the synaptic knob membrane to cause exocytosis? What has to happen? What covers the synaptic vesicles? Calcium. So how does calcium get in there? Yeah, the voltage-gated calcium channels. That's exactly what we're going to see. All right, so that nerve signal or action potential comes and triggers the opening of the voltage-gated calcium channels. Calcium enters into the cell, covers the synaptic vesicles, then triggers the synaptic vesicles to engage in exocytosis, releasing the acetylcholine into the synaptic cleft there. All right, so you can see on this page here, we're going to start to talk about, like we did with the neuron, we talked about what goes on with the neuron, right, when it's at rest. So we're going to set the table first, and then we're going to feast and dine at this table like we did with the neuron. So to set the table, we're going to talk about the characteristics of a muscle cell when it's at rest. the first thing that you'll see up here is the resting membrane potential value of a muscle cell is, learn it, memorize it, negative 90 millivolts. Okay. In the neuron, it was negative 70. In our skeletal muscle cell, it's negative 90. Okay. How do we get to that value? The same way that we got to that value in a neuron. Leak channels and the sodium potassium ion pump. Same thing. Because when we're at rest, any gated channel will be closed. Same concepts, y'all. So you're not really learning anything new here except for a new value, which is negative 90. Okay? So the same type of scenario when we're dealing with the muscle cells we did with the neuron. At rest, we have an established resting membrane potential value, and then the leak channels always open, and the sodium potassium ion pump always pumping all the time. Same thing, nothing new. Okay, are we good on that? I don't want to assume. Okay, so you can see again some of the players here. I'll zoom in. If you look up on the screen here, you can see, all right, we have these gated channels on our sarcolemma. That is the plasma membrane to our skeletal muscle cell there. All right, so we're going to have voltage-gated sodium channels and voltage-gated potassium channels. When it's at rest, take a look at the state of all of these channels shown here in the picture. Those gates are all what? Closed. That's right. So that means nothing is moving across the plasma membrane. Look at the value here, negative 90 millivolts. Our plasma membrane has the positive charge on the outside, the negative charge on the inside. Are we cool with that? All right. All right. Okay. So now we're going to go to the second part here, right? So number 2, the second phase, step, whatever you want to call it here, of a muscle contraction, which what we call excitation-contraction coupling. So essentially what we're going to do is we're going to generate, hopefully, an action potential over here at the neuromuscular junction, and it is going to transmit its nerve signal along the sarcolemma down into the T-tubules, right? And then it's going to stimulate the opening of these other gated channels that we learned about last class to release calcium into the sarcoplasm. Because remember, right now, as we're learning all this, your actin and myosin, those are the contractile proteins, what we call the myofilaments. They're responsible for the actual muscle to shorten and change length during a contraction. We saw it on the video there, right? They're not doing anything with each other. Why? Why are they not connected or interacting with each other? Regulatory proteins. The troponin-tropomyosin complex is blocking their interaction. And the only thing that can allow them to interact with one another is calcium. Okay? Calcium. So right now, calcium is stored in our sarcoplasmic reticulum. That's that tubular network that you see here on our muscle cells, or on the sarcomere here, excuse me. You can see that tubular network with the terminal cisterna, those little bulges or those end pockets on the end, all right, of the sarcoplasmic reticulum. That's where calcium is stored right now. It's not in the sarcoplasm, because if it was in the sarcoplasm, then it would bind onto the troponin, and then it would cause the tropomyosin to expose the myosin binding site on actin, and we could get the crossbridge, which can cause a muscle contraction. This will make more sense when I show you the video again, trust me. All right, so our goal here in step 2 in our excitation-contraction coupling is to get calcium out of its storage sites and into the sarcoplasm. That's our goal. Right? So that's what we're trying to do. We've got to stimulate this muscle fiber, right? So we can get our myofilaments, the actin and myosin, to slide past one another to get that shortening of the muscle cell. All right, so this is going to include a couple things. One, the EPP, which is called the end plate potential, our action potential or muscle action potential, and then finally the release of calcium from the sarcoplasmic reticulum. Those are the three events that are going to occur in this phase 2 or step 2 that we're going to learn about here. Right? And the print is really small here, but I'm going to go through and show you those three different things here. All right. And so here at the end, that's what we're going to see. We're going to see that action potential go down into the T-tubules, and then it's going to stimulate these calcium voltage sensitive channels to open up these, what we call these calcium release channels on our terminal cisterna, and it's going to allow calcium to leave this tubular structure and go out into the sarcoplasm, where our actin and myosin is waiting. All right, so let's start off with what the heck is an end plate potential? Well, I told you what the end plate was. The end plate is that region of the muscle cell that the synaptic knob kind of hovers over. And it has all of those chemically gated cation channels for sodium and potassium. If you're not quite sure, I'll show you a picture. This is it over here. This is the end plate right there. So we're going to create what we call the end plate potential. You remember graded potentials, right? Those mini kind of action potentials. So we're going to see something not really similar to that, but almost kind of the concept will be similar, which means we're going to start to open up these chemically gated channels. Sodium will enter into the cell, potassium will leave the cell, but we'll start to depolarize the cell. Keep in mind, what is going to trigger the opening of these channels? Once acetylcholine binds onto those receptors, it triggers the opening. And so sodium enters, and potassium leaves. And so remember, way more sodium enters in, it brings its positive charge with it, and it makes the inside of your cell in that area less negative. So we call that the end plate potential. And we've got to drop, right, that membrane potential. We got to get from negative 90 to our new threshold value, which I'm going to show you here in a second. All right, I so badly want to tell you, but I don't want to ruin it. All right, so are we good so far on that? So much like a graded potential, EPPs are also local, right? And they cannot open up a voltage-gated ion channel until we get to that threshold value, negative 65. I couldn't help myself. All right, sorry, I couldn't wait. All right, so that's our threshold value. We want to go from a negative 90 to a negative 65. It's a lot bigger than going from a negative 70 to negative 55. That's only a 15 millivolt difference. All right. So again, similar type of scenario that we saw in the neuron. We want to eventually open up our voltage-gated channels. And then once we get our action potential that's propagated, you can't stop it. Okay? Like pulling the trigger on the gun, the bullet's fired, you can't stop the bullet. Okay? That's what we're going to see here. So very similar type of scenario. You just want to be familiar with the names of these areas there. Okay? So the end plate potential is going to be that changing of the resting membrane potential, that membrane potential, getting it closer to our threshold value, right, through the process of depolarization. We're just going to start to have the inside of the cell becoming less negative. Less negative. Why? Because we're bringing in more positive ions. Okay. Eventually, we're going to get enough that comes in, right, and it is going to then trigger the opening of our first voltage-gated sodium channel there. So as our EPP reaches that threshold value, we get to that threshold value by the time we approach our first voltage-gated sodium channel, we get that negative 65 millivolts, boom, we've achieved our action potential. And now it's again that same scenario that we saw in the neuron, like dominoes falling into one another. One voltage-gated sodium channel will trigger the opening of the next one. And it's a sequential event like we saw before. Okay, so the next adjacent voltage-gated sodium channel opens up, which triggers the opening up of its neighbor, which triggers the opening up of its neighbor. All the while, right, this action potential, I know when we look, I'm going to come back to this slide here. I know when you look at these pictures here, right, you see the action potential going from the left side of the screen to the right side of the screen. But I want you to understand it doesn't move linearly like your car driving down the road. It moves more like throwing a rock into water, and it spreads out, right? Like you see the ripples, right? From a rock going into the water, it spreads out in all directions. That's what happens with action potentials. They spread out. They don't, yes, it looks like it's going from point A to point B, and we know why in the neuron, why it goes only in one direction, and that's because of the inactivation gate on the voltage-gated sodium channels, but it spreads out, okay? Like ripples from a rock being thrown into the water here. And the whole time, all right, these action potentials are looking for T-tubules. And you've got hundreds, if not, well, not hundreds, you got thousands of them all along the sarcolemma. And so essentially, like water going down the drain, that's what the action potential is going to do. It's going to go down the drain of the T-tubule, looking for these fellas right here, these voltage-sensitive calcium channels there. More on that in a second. All right, so again, that depolarization occurs. It's going to be propagated, all right, as it moves from adjacent voltage-gated sodium channel to the next adjacent voltage-gated sodium channel until it gets into those tubules there. So just like what we saw in the neuron, right, with depolarization happening first, what follows depolarization? You throw something up, it's eventually going to come down. So when you depolarize something, you're going to repolarize it, all right? And that's what we're going to see. The same type of scenario is going to happen here. So repolarization is going to be caused when the voltage-gated sodium channels close, right? And then the voltage-gated potassium channels open up. Potassium exits out of the cell, taking its positive charge with it and making the inside of the cell more negative. And so we can, right? You're not going to see it to that degree, but yeah, you're going to start to get to that repolarization. All right, so I still don't have it on here yet. It's coming. All right, so like I just said, voltage-gated sodium channels open first, and then they close, and then our voltage-gated potassium channels open. And again, we'll get back to that -90 resting membrane potential there. All right, but by that time, those ripples have spread out, right? And they've moved down, those active potentials have moved down T-tubules. And again, in those T-tubules, it's like the princess that's trapped in the tower. We have to rescue, all right, calcium that's trapped in the tower of the sarcoplasmic reticulum terminal cisterna there. Okay. Questions about that? Again, I'm not going to spend too much time on this because you already kind of know this concept here. Right? And we will see, all right, a small refractory period, I will mention that, after the repolarization, much like we saw in the neuron. Remember, we had the refractory period, you had the absolute refractory period in which there was no way in heck we were going to be able to cause another nerve signal to occur. And that's brief period of time. That's when the voltage-gated potassium channels are open. And then you'll have the relative refractory period, which just means we just need a stronger stimulus, all right? So that's what we're seeing here, right? This is figure 1011. So I've already discussed all these different steps here. So at this point, we're creating that end plate potential here in the motor end plate. And so if we get enough of a change in that resting membrane potential of that -90, we're looking for our threshold value of -65. If it reaches our first voltage-gated sodium channel on the sarcolemma, game on. All right, then we generate our action potential. And then these gates start opening up like dominoes falling down sequentially. And then that's followed by the opening of the potassium, voltage-gated potassium channels there. So again, this is figure 1012. This is a great, easy way to kind of do a quick summary of everything that I was just talking to you about. Right, it even shows you the threshold value. Resting membrane potential -90, threshold value of -65. And you can see here, right, phase 1 is telling you what's going on with when the muscle cells at rest. And then when we start to get our end plate potential occurring here in step 2 or phase 2, whatever you want to call it, we start to depolarize the end plate, the motor end plate. And if we don't depolarize it enough, it's that -65 threshold value, and there's a voltage gated sodium channel in that area. Boom, we trigger the opening and it takes off like a rocket ship. We get up to that +30 value. That is the stimulus that causes those voltage gated sodium channels to close. And then we see the opening of the voltage gated potassium channels opening up. And so we get that depolarization occurring there. And then we enter into our refractory period here, right? We briefly hyperpolarize the cell. And then, of course, we have our leak channels and our pumps that help get us back to that resting membrane potential value of -90. Very similar. Again, that's why I love to teach chapter 12 before I teach chapter 10. All right. Questions about that so far? We're moving along at a good pace, y'all. Not bad. Okay. A lot of material, though. Okay. So now we've got our action potential going out in all directions, 360 degrees, all over the place. And it's going to encounter T-tubules, those holes, right? Or those invaginations in your sarcolemma. If you've ever watched, it's amazing how many things just remind me of the classes that I teach. If you've ever taken a bath and then gotten out of the bathtub and let the water drain out, when it gets pretty low, you can actually see that little whirlpool over the drain. That's what I think of a T-tubule, all right? So water going down the drain is our action potentials going down the drain of the T-tubule. All right, and so that's what we're going to see. So that action potential travels down the T-tubules, and it's going to trigger the opening of the voltage-sensitive calcium channels there. And so they're going to open on the terminal cisterna of the sarcoplasmic reticulum, the opening of calcium release channels. And I love it how they named these channels, calcium release channels. They're telling you exactly what they do. They release calcium out of the sarcoplasmic terminal cisterna into your sarcoplasm. That's where the actin and myosin, bless you, is residing there. Okay? So you see, right, calcium is going to be released from the sarcoplasmic reticulum, and then it's going to move all around or diffuse throughout the sarcoplasm, looking for those regulatory proteins. So we can now cause a muscle contraction. That's our whole point. That's our whole goal this whole time. It seems like a lot. So, I mean, this is happening every time you're moving your skeletal muscle. All these events happen. That's fast, y'all. Fast. All right. Thoughts, questions about that? All right. So that's what this picture's showing you. I'll zoom in a little bit. All right. So here comes our action potential, right? Spreading out across the land of the sarcolemma. It finds the T-tubules. It goes down into the T-tubule. Eventually, it will encounter these calcium-sensitive voltage channels here, or voltage-sensitive calcium channels, and that causes them to trigger the opening of these calcium release channels. And so you'll see these calcium release channels all throughout the sarcoplasmic reticulum terminal cisterna here, because that's what essentially this structure is. It's a calcium reservoir, right? So calcium's just hanging out there. So it's going to release it out into our sarcoplasm. Now, you don't see it here on this picture here, but this is where all the sarcomeres are with the actin and myosin, because these molecules are going to float their way over to those regulatory proteins there. So what two events are linked in the physiological process that we call excitation-contraction coupling? right? That's the first event, which is the neuromuscular junction there, right? That's going to occur. And we haven't talked about the crossbridge formation or our sliding myofilaments, but we're essentially going to unite, right, the first part of the whole contraction process with the last part of the contraction process, which is the sliding myofilaments. So we'll get in, that's what we're going to cover here in a second. All right, we call it the sliding filament theory. It's not so much a theory now. In fact, I don't think anybody calls it the sliding filament theory anymore. When I was in school, when I sat where you all sat, that was a long time ago. All right, long time ago. Okay, so let's discuss, right, the last part, the cross-bridge cycling, the actual shortening, right, of the muscle sarcomere that we will see on gross inspection as a muscle contraction, the movement of the muscle there, right? So this third event is where we're going to see the binding of calcium. Thank you. Did you write your number up? Yes, you did. Thank you. Okay. So just a quick review. Let's look at, all right, this cross-bridge cycling here and what's involved. All right, so you can see here on the left side of our screen, here's our sarcomere. So when the sarcomere is at rest, which means it's not doing anything, you can see it's kind of like in its relaxed state here. But upon closer inspection, as we zoom in here to the right at that little inset box, you are going to see calcium starting just like that video I showed you. Right? Calcium is going to float along and it's going to deposit itself onto the troponin molecule, which is this globular protein here. And so when it does that, the troponin is going to shift the tropomyosin, this rope-like protein, off of the myosin binding sites that are positioned on actin. Okay, so here's the thick filament down below. That's the myosin. And then the thin filament is this actin, which this is the one I was telling you about that looks like two pearl necklaces that are just twisted around each other. So in the resting muscle cell, these myosin binding sites are covered by this rope-like protein here. So this is what we're going to do. We're going to release calcium into the sarcoplasm. It's going to bind onto the troponin there, and then it's going to move those regulatory proteins off of those binding sites. So now we're going to get our cross-bridge cycling here going on. We're going to actually get a muscle contraction. All right? So that's what this slide is basically telling you. Calcium binds onto troponin. Troponin causes tropomyosin to move off of those binding sites. And now we've exposed the myosin binding sites on actin. So now we can initiate our muscle contraction. Okay, so at this point, the only other thing that we need is ATP. Know this, folks. Two things that you need for a muscle contraction: calcium and ATP. Okay, I feel pretty confident that you all understand why we need the calcium. You need the calcium for that. There's your answer. And you're about to learn why you need the ATP. Okay, so this brings me to cross-bridge cycling. And cross-bridge cycling is a 4-step process that I'm going to go over with you here. So you know the way I like to do it. I like to do one step, show you a picture, and then we'll proceed on to the next step there. I'm trying to multitask as I teach. I can do that. Okay, so the first step that you see up there is what we call the cross-bridge formation. So this is where we're going to see the myosin attach itself to the actin. So prior to this, the myosin globular head is in what we refer to as the cocked position. Okay, so it's going to be in the cocked position. It's going to be, and it's going to, that means when we activate it, it is going to be able to move the actin past itself. So the myosin head's in the cocked position. We have the myosin binding sites exposed. We took care of that with the calcium there. And so now what we're going to see is we're going to see the myosin head extend itself out and attach onto these binding sites on actin. And when it does that, we've created what we call a crossbridge. Let me show you a picture. There you go. That's what this picture here is showing you. Okay, so you can see now what has happened, right? If you look up at the top of the screen here, you can see calcium's come on. It's exposed the myosin binding sites. The myosin head is in the cocked position. So now for the cross bridge formation, all it has to do is it'll lift up and extend itself out and attach itself onto these exposed myosin binding sites on actin. That's the first step. Real simple. We're just going to create a connection between those two proteins. Nothing really exciting there. All right. Okay. The next step is the power stroke. This is actually where we see the muscles start to get shorter. The sarcomere will shrink in size. Right. So essentially what we're going to see is the myosin head, some refer it to as swiveling, others say it pushes it past, whatever you want to use. Right. But the term for this process is called the power stroke, where literally, right, the myosin is going to pull the actin, right, a short distance towards the center of the sarcomere. What is that line at the center of the sarcomere? What do we call that? Well, the line right in the middle. What's the myosin anchor to? The M line. Yeah, I heard someone say it. The M line. Okay, the M line is at the center of the sarcomere. That's where the myosin is attached to. So essentially, the myosin molecule is going to push the actin towards the M line, towards the center of the sarcomere. And in doing so, it shortens the sarcomere here. All right, so it's going to push the actin towards the center of the sarcomere. Now, here's where ATP comes into play. If you notice, I'll come back to this slide. If you notice at this point, at step one here during the cross bridge formation, there's no ATP bound onto the myosin head. We have an ADP molecule and an inorganic phosphate molecule that are bound onto the myosin head. Because your myosin heads have two binding sites. One of those binding sites attaches onto the actin binding site there. The other one is going to allow ATP to bind onto the globular head, the myosin head there, right? So what we'll see is once, and I'll get to what happens to ATP, but after this certain phenomenon that I'll talk to you about here in a second, the release part, right, ATP is hydrolyzed or broken down into ADP and an inorganic phosphate. So at this point, we're going to see, right, as the myosin head pushes or pulls actin to the center of the sarcomere, that ADP and the inorganic phosphate pop off. Now, mind you, myosin is still attached onto actin. I'll explain the significance of that here in a second. Okay, so that's the last point here at the bottom of the screen. ADP and the inorganic phosphate are released during the power stroke. I'll show you the video, all right? And so once that happens, now that ATP binding site is available for another ATP molecule to come on. Here's where it's significant about ATP. We need ATP to give us the energy to detach the myosin globular head from the actin. So we call that the release. So once ATP binds onto that available ATP binding site on myosin, it causes the globular head of myosin to disconnect or release from actin. And so two things can happen. We can either go to rest and not have another muscle contraction, or we can do another muscle contraction. All right. But we need that ATP to give us the energy to release that myosin head from actin. Anyone here ever hear of rigor mortis? That's what's responsible for rigor mortis. When you're dead, all right, your cells aren't making ATP anymore. And what did I say? What do you need for a muscle contraction? You need calcium and ATP. The ATP, I just told you, we need that ATP for the myosin to disengage or release from the actin. Well, if it can't disengage, right, the muscles will feel stiff. Now, rigor mortis only sets in for a certain period of time because, like with anything, when you die, your tissues start to rot and degenerate. Well, these proteins will do the same thing. They'll rot and degenerate, and then they'll disconnect on their own. Okay, so rigor mortis is not forever. Okay, but it's usually, initially after a person dies. I can't remember how long. It varies. You'd have to ask a forensic person that deals with all that, or a mortician. They would probably know better than I. Okay, all right. So that's the third step, right? The release of the myosin head. That's what we're going to see here. All right, so in order for that myosin head to release from actin, ATP jumps onto that myosin head, and when it does, it pops off, leaving us the last step, which is where we're going to reset this whole phenomenon to either rest the muscle or do another muscle contraction. So that leads us to step 4, and that's the reset. So we're going to reset. In order to reset the myosin head, here's how we do it. ATP is going to undergo a chemical reaction called hydrolysis. And so essentially, what is that? That's when we split that ATP molecule into ADP and an inorganic phosphate. And the enzyme that helps to catalyze this is called ATPase. Right, so that's the enzyme that we use to catalyze, to initiate, to help with this reaction of breaking down ATP into ADP. And so that's why you saw at the beginning here of that Crossbridge formation. Well, hold on, let me show you this slide here. All right, so in order to reset or cock the myosin head back into the ready position, we hydrolyze that ATP into ADP in the inorganic phosphate, which is still attached onto the ATP binding site. So that's why when we looked at the Crossbridge formation step here, that's why you have the ADP and the inorganic phosphate still attached onto the ATP binding site. It just got done resetting the myosin head for another contraction. And when you see it in the video, it'll make more sense. This is usually the part where I ask, any questions? And people don't say anything because it's still a lot to absorb. Don't worry. You have this video here that can help you. Yes, ma'am.
But how long does that look like?
Oh, instantaneous. I shouldn't say instantaneous. Probably 1 to 2 milliseconds is fast. It's fast. All right. So here you can kind of see when we're looking at our overall sarcomere appearance. If you look up here, that's the relaxed sarcomere. Okay, no cross-bridge formation occurring. And then here's what it looks like in the center of our screen here. This is what the sarcomere looks like during that cross-bridge cycling there. So you can see it got shorter. See here, H zone looks larger. We get down here into the crossbridge cycling sarcomere, right, virtually disappeared. Okay, so the distance from this Z line to this Z line has become shorter when we get crossbridge cycling. Look at the distance for this Z line to that Z line. It's a lot larger. And that's because the myosin molecule here, proteins, pull the actin towards the M line. Okay. And before we break for lab, I'll just show you that video, that 4.5-minute video there. Okay, so cross-bridge cycling can, and again, you'll see this probably again on a test, As long as you have calcium available to keep the regulatory proteins away, and as long as ATP is present, we can continue on doing the crossbridge cycling. Right? And so as a result, like I said, we shorten the distance between those two Z discs there. And so last class, we learned about the H zone and the I band, how those will get smaller and smaller and could potentially disappear during a maximal muscle contraction. All right. Another test question too, you might see like a true or false, right? Do the thick and thin filaments ever change length? No, they never change length during this whole process. The spacing between them do, right? But they don't change length. It's a great true false question. All right, so here, this slide here shows you muscle at rest, and then this person just starts to bend their elbow, and then the sarcomere shortens there. And so you can see it here on the electron microscope, the two Z lines here, the M line in the center, and then you can see the distance between these two Z lines, or Z discs, sorry, as they get closer to the M line in the middle there. Cool? Cool. All right, so what is the function of calcium in skeletal muscle contraction? I feel like now everybody can tell me. What is it? To keep the baddies away, to keep the regulatory proteins away from the binding sites, y'all. All right, that's a long explanation, but that's essentially what it is right there. That's why we need calcium, right? It keeps, all right, tropomyosin off of the myosin binding sites on actin so we can get our crossbridge cycle. Right, so it's like garlic to a vampire, all right? So what causes the release of the myosin head from actin? What attaches on to the myosin head? ATP, yep. So ATP binds on, that allows the myosin head to disengage, and then to reset it, okay, we're going to use that energy, all right, from the when we hydrolyze ATP into ADP in the inorganic phosphase. And so what allows us to do that is this awesome enzyme here called ATPase. Okay. So our next clinical view, muscular paralysis and neurotoxins, fun stuff. I'm sure some of you have heard of both. We were talking about Botox and botulism before, but to kind of revisit it. So tetanus. So we have two, all right, microorganisms. All right, we have Clostridium tetani and then Clostridium botulinum. Anyone here take micro? Anyone here has to take micro? So you have taken it or you will be taking it? Okay. Would you say it one more time? You have to. Okay. So you learn about these. All right, and for those of you that haven't, don't worry. If you're going to be taking 211, most of my students that are taking 211 also take micro. And you learn quite a good amount. But so when we're talking about tetanus, we're actually talking about the toxin that Clostridium tetani releases here. And so it is an inhibitory neurotransmitter. Well, gosh darn it. We learned about inhibitory neurotransmitters when we were talking about IPSPs. Remember those inhibitory, all right, postsynaptic potentials? So this toxin is going to act as an inhibitory neurotransmitter. So its job is to prevent the creation, all right, of action potentials. And so that's what it's going to do, right? And so that inhibitory neurotransmitter, if we take that off the board, what's left? Only excitatory neurotransmitters. So what do excitatory neurotransmitters do? They excite the neurons, which will then stimulate the effectors. And in this case, it's going to stimulate skeletal muscle cells to what? Contract. And that's what we see here. We're going to see an overstimulation of those muscles. Now, the famous thing that folks will refer to with tetanus is lockjaw, right? It causes, and we already know what this muscle is, the masseter muscle, right? In extreme cases, it causes it to contract and it won't stop contracting. You can't literally open up your mouth, right? But it's not just for the masseter, it could be wherever, right? And it can potentially be, like it says up here, life-threatening. So that's why, you should get your DTaP. I think it was every 10 years for the booster for the DTaP. That's the one I don't like because my arm feels like someone punched me in it, repeatedly for an hour because it's like sore for 3 days afterwards. So that's tetanus. All right. Botulism comes from Clostridium botulinum. Again, another toxin. So tetanus causes what we refer to as spastic paralysis, which means the muscle just goes into muscle spasm and it contracts and contracts and contracts. Botulism, causes our muscular paralysis, but it's more like a relaxation version, meaning we can't get a muscle contraction out of that. Okay, so you want to know the difference. One causes the muscle to contract incessantly. The other one causes it to almost be paralyzed where it won't contract there. That's why we use it for Botox, because it literally paralyzes those muscles, not to the point where you can't use them at all, right? But they'll relax and it takes the wrinkles away here. So how does it work? If we can't release our acetylcholine at the synaptic knobs, we can't generate an end plate potential. We can't generate an action potential, a muscular action potential. We won't release calcium from the sarcoplasmic reticulum there. All right. All right. So understand that we can use this to treat folks with muscle spasms. And this is that story I was telling you about, that one patient whose neck or head was literally resting on their shoulder because their muscles were in such bad spasm. And you'll see it in a lot of folks. All right. I've completely changed my take on botulism. All right. On Botox. All right. This is a great slide here. I think we have a few more minutes. We do. All right. I will encourage you folks to really understand, right, the events of a muscle contraction, which is pretty much what we just did. This slide now pretty much talks about what we just learned in reverse, right? So essentially that's what we're going to see here. So let's go through it step by step. Events in the muscle relaxation. So to start off, we're going to shut our motor neuron off. Bless you. So we're going to stop with the nerve signal coming down our motor neuron, which is going to prevent the release of our neurotransmitter because we're not going to open up those voltage-gated calcium channels. So calcium won't enter into the cell, and the synaptic vesicles won't release the neurotransmitter. So we're going to prevent the acetylcholine from being released from our motor neuron. So any acetylcholine that's been released previously that's in the synaptic cleft and that's bound to those cation channels, those chemically gated cation channels, we're going to have our acetylcholinesterase enzyme out there break all that stuff down. That chemical process of breaking down acetylcholine into acetyl and choline molecules is called hydrolysis. All right. So now we've just gotten rid of all the acetylcholine that was in the synaptic cleft. That causes, right, those gates that were once open, those chemically gated cation gates, to close down. And so now we're no longer going to have an end plate potential or an EPP. If we don't have an end plate potential, we're not going to have a muscular or a muscle end plate potential being generated along the sarcolemma. And if we don't have that going on, that means we won't be able to open up. We will, in fact, close our voltage-sensitive calcium channels, and we won't then open, or we'll close our calcium release channels on the sarcoplasmic reticulum terminal cisterna. And so the only thing that's going to be operating at that point are going to be the calcium ion pumps. And what's their job in the muscles? To pump, I mentioned this last class, to pump calcium from the sarcoplasm into the sarcoplasmic reticulum terminal cisterna. That's what it does. That's what the calcium ion pumps do in our muscle cells. They pump calcium out of the sarcoplasm. Well, guess what? If we don't have calcium in the sarcoplasm, what's that going to do to our regulatory proteins? Troponin will then cause tropomyosin to shift over the myosin binding sites. And now we've blocked those myosin binding sites. So the muscle relaxes, and due to one of the many characteristics that muscle has, one of that being elasticity, it causes the muscle to rebound back into its original position. Kind of see that? It's kind of like everything in reverse that we just talked about. So I would say, learn this slide, memorize this slide if you can, all right, if you have time to do that. Time. All right, how do choline, acetylcholine esterase, and calcium pumps function in the relaxation of muscle? There you go. So that enzyme is going to continuously break down acetylcholine, right, into its individual components. And then the neuron is going to vacuum all that stuff back up into the synaptic knob. And then the calcium ion pumps are going to pump calcium out of the sarcoplasm back into, all right, the sarcoplasmic reticulum, terminal cisterna there. So there's no calcium available. And so then the regulatory proteins block the myosin binding sites on actin. Pretty much everything I just talked about. Cool. This will probably be a good break. Yes, this is a perfect time to take a break. Okay.